Hello, honors biology students. It's great to have you back for our second full week of distance learning. So today we're going to look at Punnett squares and just give a brief overview of that. This picture here was sent to me by Kate Black. She's at her house studying with her fur baby, Bailey. So thanks for sending that to me, Kate. Feel free, guys, to send me pictures and I'll try to incorporate them into the slides. Okay. All right, this is on the website as well. I posted it. Words for geneticists to live by. Remember, nobody walks around haploid. We have pairs of chromosomes. We're diploid organisms. And then use the same letter for the same trait when you're doing test crosses. So when we talk about the same letter for the same trait, that's talking about using, capital B and lowercase b. Capital B would be dominant alleles and lowercase b would be the recessive alleles. We wouldn't use a B and an R for the same trait. Like if you're looking at brown hair and red hair, you would pick one letter. Do not use two different letters. That's all that's referring to. Okay, so Punnett squares. So some of you guys did Punnett squares in middle school, some of you did not. We're going to take this slow this week. This is all we are doing this week. So I know on the pacing guide, it might look a little overwhelming. The worksheets, you're like, giant worksheet. It walks you through things slowly. I am here for questions. This is all we're doing this week is just focusing on monohybrid Punnett squares. That's like all we're doing, okay? So we're going to do this. We'll get through it. Okay, so Punnett square, honestly, I think these are fun. They look kind of like puzzles, okay? So the Punnett square has these 4 boxes. This one is for, if it has the 4 boxes, it's a monohybrid cross. Mono means one. That means we're looking at one trait, right? Next week, we'll do dihybrid crosses, but this week we're focusing on monohybrid, okay? So it helps us predict the different genotypes resulting from a cross from 2 different parents, okay? Which also, looking at this, we'll be able to tell the phenotype. So we got genotypes and phenotypes. So we have the possible gametes, so the alleles that the parents could donate. So in this case, these are both heterozygous parents, right? They have one dominant allele and one recessive allele. And we can see on this cross, what happens is you bring these letters down and over to give you the possible genotypes that go in the different squares. So this one, you bring this one down, you'd bring this one over, right? This one, you bring this capital A down, bring the lowercase a over, right? They're kind of fun. All right, so in this case, when you have the 2 heterozygous parents, we have actually 3 different types of genotypes. We have a homozygous dominant, we have 2 heterozygous, and we have 1 homozygous recessive. So the boxes are showing the possible genotypes of these parents, okay? So it gives us ratios of the genotypes or phenotypes. So when you're asked for a ratio, it's either going to ask you for a percentage or you can do, I'll show you what a ratio means. Okay, so for this particular square, we have 1 homozygous dominant, we have 2 heterozygous, and we have 1 homozygous recessive. So we'd have a 1 to 2 to 1 ratio. If I asked what the percentage was of homozygous dominant, what's the chance of that happening? You could say 25%, right? 25% because we have 4 squares. And so if we fill up one of those, I didn't mean to do that, that means that we have a 25% chance of having this homozygous dominant, okay? You can think of this whole box, these 4 boxes, you can think of it as a dollar bill. If you struggle a little bit with percentages or ratios, you can think of each of these boxes like a quarter, right? 4 quarters equals $1. So if you fill up one square, that's 25 cents, another square, that's another 25 cents, another square, that's another 25 cents. So for the phenotype, which is the physical characteristic of these 3 with the dominant allele, you have 25% plus 25% plus 25%. So you have a 75% chance, 75, this drawing tool is so amazing, wouldn't you guys agree? A 75% chance of the dominant phenotype, okay, whichever that is on this. All right, so this is an overview of the different generations. This slide is probably going to mean nothing to you right now. You really need to work through the different generations, the different practice problems that we're going to do, and then this will make more sense. So I'm going to walk through it, but if it doesn't make sense right now, that's okay. You kind of need to do the Punnett squares yourself, the test problems for it to make sense. So this is a parent generation that has pure parental plants. Okay, we're talking about plants because it says over here purple and white. So we're going to assume we're talking about pea plants. So if you have a pure purple pea plant, that means you have two of the same allele. So you have two capital letters because it's a pure purple plant. And then if it says a white plant, that means you'd have homozygous recessive, you'd have two lowercase alleles. Now, a white plant is always going to be pure anyway, because you have to have the two recessive, the two lowercase alleles for it to be white. Okay, but for the purple one, it does matter if it says pure or if it just says purple or if it says heterozygous. So you have to pay attention to that. If you cross these two pure parents, you're going to get in that first generation, after you cross these, their babies are all going to be heterozygous and all their phenotypes are going to be the same, right? Because they all have that dominant allele. If you cross these two heterozygous in the next one, you're going to get some really interesting information, some interesting ratios. You actually saw this in the last slide. That would be this one. This would be an example of two heterozygous parents. Your genotype, which is these letters, would be 1 to 2 to 1. Your phenotype, though, is going to be 3 to 1, the physical characteristic of the trait. This F3 generation, this would be an example of if you did a test cross, you don't know when you cross two of these plants together, you don't know if one of them is homozygous dominant or one is heterozygous. So this F3 could be considered a test cross. We'll talk about that more later. So this is just, again, reviewing what I just said. The P1, this would be pure parents. So again, homozygous parents. So you have homozygous dominant, homozygous recessive. All of the children of this cross are going to be heterozygous, and they're all going to have that same phenotype. They all have the same physical characteristics. They're all going to look the same, okay? For the next one, if we're crossing two heterozygous parents, which means they have one dominant allele and one recessive allele, you're going to get that ratio of 1 to 2 to 1. One homozygous dominant, two heterozygous, and one homozygous recessive. And then our phenotypic ratio, which is the physical characteristics, you can see here, this makes it a little more obvious, is going to be 3 to 1. So we're going to have 3 purple plants to 1 white plant. So F2 would be the test cross of, say, a purple plant from that last generation. We wouldn't necessarily know right away if it's homozygous dominant or heterozygous. If you get white flowers in this cross, then you know that this parent is heterozygous. Because think about it, this parent has to donate one allele, right? It has to donate one. If it only has dominant alleles to donate, then all of the children are going to be purple because they'll all have at least one dominant allele because you get one from one parent and one from another. And if one parent can only give you dominant alleles, then that's the trait that's always going to show up in your phenotype as your phenotype. Okay, so I think that's where we're going to stop today, actually. So I am going to give you a practice problem. We're going to go through it. So I'm going to go ahead and do another video with that. But this was just an overview of Punnett squares that I hope you enjoyed.